So I'm Simone Booker at Dalhousie University, and I just want to say hello to everybody and welcome to this week's SEDS online webinar. As always, we'd like to thank our sponsors from IAS, which allows us to offer all these resources free of charge. This includes recorded lectures, learning tools, and even virtual field trips. So take a look at the website to see all of the new info. Today's lecture is by Dr. Hilary Corlett, who is an assistant professor at MacEwan University in Edmonton, Canada. She got her bachelor's degree at Queen's University and her master's and PhD degrees at the University of Alberta. Hilary has an interest in the links between the terrestrial and the marine environment in the Paleozoic and the present, as well as early and burial carbonate and evaporate diagenesis and chemostratigraphy. So please let's welcome Hilary in her presentation, Hyperspectral Imaging in Sedimentary Rocks at Core and Outcrop Scale.
Thank you, Simone. Welcome everyone, and thank you for attending today's lecture. I'm going to be running through two different examples, one at core scale and one at outcrop scale, of the application of hyperspectral imaging. I figured this might be an interesting topic for the SEDS online seminars because it applies to both siliciclastics and carbonates alike. And I've learned quite a bit over the last few years working with a lab at the University of Alberta, the Spectral Imaging Lab, with Dr. Benoit Rivard and Dr. Jialou Feng on using this technique. And I found it to be just incredibly helpful. So I hope that this is helpful for you as well. Okay. So first off, I'm just going to explain a little bit about hyperspectral imaging. What you might be used to in terms of satellite imagery would be something more like multispectral data. So things like spot data or quickbird data. This images within 3 to 10 bands of the EM spectrum. So the image down in the bottom left shows a typical kind of satellite image that you might see. Now, it has a lot of information in it that you can use for classification of vegetation and various things like that. But in terms of the bands that it encapsulates, it's disconnected 3 to 10 bands, not continuous. And in hyperspectral data, we're looking at continuous bands. You can have hundreds of bands that are captured depending on what sensors you have to detect those bands. So you can detect in the shortwave infrared range or the longwave infrared range, and it's continuous data, so they aren't separated into those bands. And you can see both techniques are much more extensive than what we see in the visible infrared range, so what we just see with our eyes. This is the lab that I've been working with. So this lab is run by Dr. Benoit Rivard at the University of Alberta. MacEwan and Alberta are both in Edmonton, across the river from each other, so it's pretty easy to work together. And this spectral imaging facility has 2 core scanners that look like this. These are Sisu rock scanners. This one is the shortwave infrared, and there's also a thermal infrared just beside it that images in the longwave infrared range. They also have a handheld spectrometer and an outcrop scanner. So I'll be talking today about the drill core scanner with some examples from some organic rich black shales. And then the outcrop scanner will look at carbonates in the Cathedral Formation, which is a middle Cambrian formation exposed in the Rockies in Canada. So first off, just to introduce the shale that we're going to be looking at, it's the Duvernay Formation, which is an Upper Devonian Franian organic rich mudstone and shale. And if we take a look at this map here, so just for anyone who's unfamiliar, Alberta, Canada is located right here. And the interval that we're going to be looking at comes from a core that was drilled in this region over here. The blue on this map shows the Leduc Formation, which are Devonian reefs equivalent in age to the Duvernay Formation. And the Duvernay Formation basically is equivalent to the first 2 intervals of reef growth in the Leduc. In this particular area, this is the K-Bob field. It's currently actively being fracked and produced out of. The base is clay rich, then it's overlain by a carbonate that is intercalated with thinner clay beds. And the upper Duvernay is actually quite silicious. So it's more of a mud rock than a true shale. So up above is an image of the entire Duvernay core, including some carbonates from the Beaver Hill Lake group down in the lower end, which is on the left-hand side, and the Ireton shales capping them off at the right-hand side. Hyperspectral imaging is extremely useful, especially in shales, I find, for analyzing core. It's extremely fast. You can scan 100 core boxes in about 4 hours. There is some processing that will take place after the fact, but that's something you can do, wherever the data goes to. So you don't necessarily have to be in the lab for that. You just have to have the proper software. In terms of collection of data, though, if you have a limited amount of time with a certain core, it's a great way to get a ton of information out of it very quickly. You can obtain continuous mineral and TOC data. It is non-destructive, so you are not, if a lot of the time in core labs, they will say, you can't sample in certain intervals. They want to preserve the core for as long as possible, which is great. So this is a technique that allows you to analyze many, many things without actually taking any samples. But if you do need to take samples, it's also a way to highlight all the heterogeneity in that core in order to appropriately sample it for what your task is. You can decipher biogenic structures, sedimentary structures that might not be visible with the naked eye. And another really interesting thing with long-wave infrared data, you can actually decipher between amorphous silica that may be biogenically derived versus a crystalline silica. So here's a look at that same core, but imaged in the shortwave infrared and thermal infrared or long-wave infrared range. And in the shortwave infrared range, the CC rock scanner is able to image at a resolution of 0.8 millimeters in every pixel. At a normal infrared range, this would be for scanning fairly rapidly. That would be the 100 cores in 4 hours, 100 core boxes in 4 hours. If you needed higher resolution data, you can actually move the stage and image at 0.2 millimeters per pixel for a higher resolution image. The long wave infrared scanner is not at the same resolution. It's a little bit less, so it's getting close to being a millimeter per pixel, 0.83. And you can see from the imagery immediately, what these are is the shortwave infrared and thermal infrared. They have been basically processed in this case just by Rgb imaging, where we just are almost like false coloring. You're taking the RGB bands and just positioning them at certain intervals in the spectrum that will highlight the greatest variability that's possible in the core. So in terms of shales, these intervals where we've highlighted the red, green, and blue, this is just basically positioned at intervals that work very well in shales. This would maybe be different if you were dealing with something like a carbonate core. You can see in the shortwave infrared, even from this tiny picture here, where the core is, obviously there's quite a lot of core there, that you can immediately see differences in the overall mineralogy as you go through the core, but also that certain structures are showing up. So I'm just gonna talk a little bit about the differences in mineralogy that you can see. So here's an image a little closer up of one of these core boxes in the Duvernay. And once we process the imagery that's collected through the scan, we have the shortwave infrared image on the the left-hand side here, and the long wave in the right-hand side. And in the shortwave infrared range, we're looking for clay minerals and carbonates are active in this range within 100 to 2500 nanometers. And the long-wave infrared range will also provide data on carbonate and shale, but it's particularly useful for differentiation of the different quartz species. So this graph down here from Rivard et al. 2018, which has a number of very helpful references in terms of the history of how this technique has been applied in sedimentary rocks and other deposits like kimberlites and things like that. This reflectance spectra here is showing basically a typical carbonate spectra, a typical mudstone that might be intercalated with carbonates, and then TOC-rich shales. So each of these is very easily differentiated between, and then you can actually pick out certain parts of the spectra that would be highlighting just a particular type of clay or just a particular species of carbonate, for example, or phase of carbonate. One thing I've just highlighted here is an interval where you can see that this has actually highlighted this region just in this zone right here. I hope you can see my pointer, otherwise this is not going to make a whole lot of sense. But what's highlighted in the box there basically is showing this interval here that has some pyrite in it. And actually sulfides on their own, they would image, you wouldn't usually be able to pick them up in the imagery. So for certain things, it's not going to be able to detect them, but for most things in shales, it's going to be particularly useful because as you can see from looking at the core box, very little is visible in this core, just looking at it. And logging a core like this takes an extreme amount of patience and time in order to recognize tiny little biogenic structures, or in terms of changes in mineralogy, it's almost impossible to detect them. And you would have to go ahead and just sample at regularly spaced intervals without having any idea whether you're capturing the heterogeneity at a proper sampling interval. So here's another look at that same core. I just wanted to show how it compares to changes that we see in elemental data. So first off, this is our Rgb image that just highlights the differences in mineralogy within the core. And just the depths are located along the left-hand side there. If we take a look at some data that was collected and analyzed, this is from spot samples that were taken at regularly spaced intervals. Oh, sorry. No, this is my XRF data that I took at every 2 centimeters down the core. And at the right-hand side, we have several clay proxies, and we have carbonate and silica. I have my zoom in the way of the screen here, so I can't actually, I think this one's iron. The green one's iron. And basically all of these are showing the low concentrations to the left-hand side and the high concentrations to the right-hand side. And the image there is one just in general looking at clay. So not a specific type of clay, but just overall the clay content in this particular interval. So we can see that actually the lower portion of that core is very clay rich and the upper portion is more carbonate rich. But from looking at the image before, we can, it's not immediately obvious. It's definitely not obvious when we look at this. We can see a slight change from a lighter gray to a darker gray, but it's not very telling. So in terms of understanding whether this core should be sampled at a meter interval or a half meter interval, it's very helpful to have this type of data that is immediately able to tell you what the heterogeneity is like in the core. Another really interesting thing that that this technique is capable of doing is to actually detect levels of TOC. So in this example, this is from the Rivard et al. 2018 paper. And in this, they've been using this technique to look at a number of cores in northeastern BC, British Columbia. And these shale intervals, the EV, Otter Park, and Musqueam, all have variable levels of TOC. And these were able to be detected through a wavelet analysis where they're basically looking at indicators in the core that would correlate to TOC, and then using those indicators in the spectra to detect the TOC. So it can't directly detect the TOC, but there are certain spectral features that are associated with the TOC that can be used to actually determine the level of TOC. And in terms of the correlation, you can see the predicted versus the actual observed weight percent. So this would be an analysis that has been done and you would do in any kind of detection of TOC or minerals. You usually want to try and grab some samples in order to verify that you've picked the correct spectra, that the technique is working. And in this case, the TOC model has been fairly successful in detecting the weight percent TOC. In terms of looking for biogenic structures, in this core, this is the middle carbonate in the Duvernay. This is a carbonate wedge that's basically being deposited from east to west in the basin, and it thins out westwards. It's an interval that causes quite a lot of problems for production in the Duvernay Formation. So fracking in the carbonate in this region is something that they try to avoid. And it's fairly easy, obviously, to detect this interval in well logs, using well logs. So that's not really an issue. In terms of us figuring out how this carbonate got there and what's happened to the carbonate, it's obviously pretty hard to see anything here, and you would just assume that it's been deposited and that's the end of the story. But when you actually take a look at the spectral imagery here, so this is shortwave infrared spectral imagery, and we can see that in this exact same interval, there's actually several biogenic structures that are completely not visible. And that's not just because my photo's a little bit blurry. I couldn't see these things in this interval at all. So all of a sudden you have more information in terms of the timing between these intercalated thin clay-rich and carbonate beds. You can understand what's gone on in between. So obviously there's been a bit of a lag between the carbonate being deposited versus the clay, because there was enough time for the carbonate to be bioturbated, or the carbonate was bioturbated and infilled by that shale. We can also see two different types of burrows. So up in the clay-rich beds, it's more of these horizontal burrows, and in the carbonate, we're actually seeing more vertical or subvertical burrows. So just another look here. When you look at that's the Rgb image that I was showing you earlier, but you can actually then also from the same data set pull out the carbonate spectra and see where you have the highest level of carbonate versus the distribution of clays. And then you're able to also tell what's infilling the burrows definitively. And we can see that the burrows are infilled by organic rich material, essentially. The Duvernay core that we're looking at, this is the well log here on the left-hand side. We can see that middle carbonate unit, and also what I'm going to show in this plot on the right-hand side is a plot of, this says silica versus silica. Sorry, that's silica on the y-axis and aluminum on the x-axis there. So this is a silica and aluminum plot. I mentioned earlier that the upper Duvernay is a siliceous mud rock in the above that carbonate unit. And what we see in the silica aluminum plot is actually that we have this region of excess silica. And this is defined by Rao et al. And others who work in shales, Jürgen Scheiber. There's a lot of early 2000s work looking at evaluating the silica versus aluminum ratios to essentially figure out whether the silica that is in your shales can be attributed to a biogenic source potentially, or whether it's all attributed to clays. So in this plot, we have the several different things that are informative in terms of what's in this core. So we can see that there's a zone where both silica and aluminum is very low. This would be the carbonate dilution zone. So this is where we have more enriched carbonate. Then anything that falls outside of the silica excess and the and the carbonate dilution zone, this is actually clays. And so in a way, this can help us immediately differentiate all the different zones in the core. But of course, sampling all of this was done actually prior to the hyperspectral analysis. We sampled at half-minute intervals, and the whole sampling and analysis process took an extremely long time and was also very costly. So we had, all of our samples run through ICP-MS and OES, and that can certainly take a lot of time and money. Of course, it tells us a lot about the core, and having that really high excess silica is quite interesting. What's causing that? Is it biogenic source or is it detrital? Of course, we could have a biogenic source from something like radiolaria, which were around in the Devonian, and possibly an indicator of upwelling. Or it could actually be just quartz silt. So it doesn't necessarily have to be a biogenic source. This is a look at that upper Duvernay interval just in thin section. And what we can see is that there's these tiny little, could be quartz grains, could be not quartz grains. There's not actually a whole lot of quartz visible in any of the thin sections in this interval. So, right away that might be a clue that we don't have a lot of quartz silts, but in fact, we might have the biogenically sourced quartz. And the great thing with using the longwave imagery is that you can actually detect the difference between amorphous and crystalline silica. Now, these cores are at 3 or 4 kilometers depth. So actually, you would think that opal A or CT might have, at this point, gone through silica diagenesis to the point where it's a crystalline quartz. But in silica diagenesis, it can actually be slowed through the presence of clays. It changes the rate at which silica diagenesis occurs. So in fact, in the Duvernay core in the upper Duvernay, we see a silica that is still amorphous. It's more of an opal CT. Just having a look at the spectra here, this is a hypothetical plot of different quartz spectra. We have basically crystalline quartz displaying a doublet reflectance, 2 peaks at 8,400 nanometers and 9,200 nanometers. This is something that's caused by the crystalline structure of the silicon tetrahedra and basically how they're assembled. The stretching of the SiO-Si bond causes this doublet peak. In amorphous silica, we actually see that there's just a single peak at 9,000 nanometers. And what we've seen through imaging various samples from museums and different labs is that there is a change from the single to the double peak, where we see a shoulder developing around the 8,400 nanometer range that would possibly indicate that you can detect the silica ordering, basically, and the silica diagenesis, the different stages of silica diagenesis using this technique. This hasn't completely been verified, so I'm not going to, that's why I have hypothetical up here. This is just from preliminary work that we've done, but extremely promising in terms of definitely just being able to detect whether you have amorphous silica or whether you have a crystalline quartz. And in this case, in the Duvernay, we definitely see that there is quite a lot of amorphous quartz in the upper Duvernay. So here's the imagery with a grayscale. This is just showing the level of, the amount or abundance of that single amorphous peak. So we can see that in the upper Duvernay, there is an enrichment in that amorphous silica. And the spectral plots that are just above that are the actual ones that came out of this image. And the red peak there is the is the single peak for amorphous silica at 9,000 nanometers. And then we can see the doublet, which is the green peak, double peak. So through scanning the core, we were able to detect all this continuous mineralogical data. And we were also able to differentiate between these different quartz species. Now, just to check, basically, you want to have multiple techniques to check all of this information. We actually took some samples from the Upper Duvernay and ran them through XRD, which is also capable of differentiating an amorphous versus a crystalline quartz. This is the test, basically, that Murata and Norman recommend for determining from XRD data whether you have an amorphous or crystalline quartz. And essentially, the crystallinity index shows that a fully crystalline quartz would be a 10, and an amorphous quartz would be a 1 or less than 1. In this case, the crystallinity index is 7. And of course, this was just a bulk sample. So in terms of actually being able to pick out the exact interval of where we have amorphous silica versus not, we weren't able to do that. So overall, this is showing sort of a mix, possibly, of what we see in the spectra, which looks more from the single peak like a pure amorphous silica versus a crystalline silica. So we might have both in here, but overall, we don't have any quartz that is entirely crystalline in terms of what was analyzed or what the XRD analysis showed. Okay, so that covers kind of what I was going to talk about in terms of this technique used on cores. And of course, you might think, okay, well, I don't know anyone who has a core scanner, for example. You can also get spectral data from using a hand-held device. The difference is that it's not paired with high-resolution digital imagery. So the imaging is very helpful in terms of the visual that it can get you, but you can get the same records in terms of continuous mineralogical record or semi-continuous by using a hand sampler. Okay, so the next thing I wanted to show you, just because I wanted to try and cover the gamut here and show the overall utility of this technique. So I'm going to show one example of an outcrop scan that my research group did, but I'll start by just highlighting some studies, if you're interested, on other outcrops that have been scanned, and these are carbonate outcrops. So Tobias Kurtz in 2012 published this paper in Sedimentology that shows the hyperspectral imaging of a quarry wall. So this is in Spain, and the wall is, fairly pristine. You can see that it's very flat. There's not going to be a whole lot of shadows that are going to interfere in collecting the data. And they were obviously able to be quite close while scanning. So this is kind of an optimal situation here, where you have a very, almost like a thin section face to image. And in the next example, in this example from Central Oman, this was published in 2018. And we can see that they did this from a distance of 50 to 300 meters away from this cliff. And within the paper, they also show the results of scans that were at much greater distances. They did determine that you're not able to detect very small scale variation unless you are at a closer distance. But overall, you can use things like a carbonate wavelength image to actually image the difference, image boundaries between limestone and dolomite, for example. So in our example, we were working in this area for a couple of students that Professor Kathy Hollis and myself work with on some Middle Cambrian Dolomites. So just to set the scene here, we're in Alberta, and we're right at the Alberta-BC border within this box here. This is the David Thompson Highway, and it runs through an area that actually sits between Jasper and Banff National Parks. And there is a whole host of Middle Cambrian outcrops in this region, and some of them are spectacularly dolomitized. This is a pretty famous outcrop in terms of, if if you've ever visited Canada and gone on a hydrothermal dolomite trip, you might have actually seen this outcrop. We're sitting just here, and oops, sorry, Whirlpool Point would be just in this region here. And we have our setup, which is basically a truck, very common in Alberta, giant trucks, and the imaging sensors over here. So our source of light in this case is just sunlight. So it's best to have a sunny day with no clouds. And we have the truck in order to move the equipment along, so we didn't have to keep carrying it, which was quite handy. And we imaged roughly 250 meters along this outcrop. I think I have the exact number in a second. So here's a look at the outcrop just a little bit closer in. This is an outcrop that has undergone extremely high temperature diagenesis. So up in the range of, these settled dolomites are in the range of 200 degrees C. And the brecciation and zebra-dolomitization that you can see in this outcrop is extensive throughout the outcrop. In this region, there are also the carbonates have also undergone an initial dolomitization phase with replacement dolomite. So all of the ridges that extend along the mountain ranges in this region will display stratabound and non-stratabound dolomites. And in this particular outcrop, we just got lucky in the fact that the highway had to blast through this part and expose this amazing breccia. The breccias aren't quite as common in the region, but there are other locations that you can see, for example, the zebra dolomites. And the replacement dolomitization is visible in all of the cliff faces. So we're sitting just here in the middle Cambrian in the Cathedral Formation, and we are looking at this breccia. Here's just a look at the outcrop itself with a little bit more information. We've got basically a brecciated zone in the hanging wall of the fault, which is much larger than in the footwall. You can see a smaller brecciated zone here. Some replacement dolomite, so dolomite that is not displaying any of the brecciation or any of the zebra dolomitization. Away from the brecciated zone, we see the zebra dolomites extend quite a long ways. And from looking at the outcrop, it was already obvious that there were potentially going to be several phases of dolomitization that this area had undergone, just based on the colors we could actually see with the naked eye. Overall, first of all, I'm going to show you a wavelength image. So in this type of imagery, what we're looking at is the ability to detect the difference between dolomite and calcite. So this would focus in on the carbonate wavelength and that range, and basically differentiate between the more magnesium-rich and the more calcium-rich wavelengths. And of the whole outcrop, 25 meters. This is just 25 meters here. The whole outcrop is about 200 meters. If we take a look at the image here, this is the entire section that we scanned. And we have the contact between dolomite and limestone just at the very far right side of the screen. So this contact was not necessarily visually that easy to pick out at the outcrop. We had to test with acid as we went along to try and find this. But with the wavelength imagery, it was able to detect that boundary and show us how diffuse that boundary is. And it in fact looks like it's fairly sharp. We see in the associated petrography that mine and Kathy's student, Jack Stacy, did some petrography on these samples. And we can see that there's a limestone contact, again, fairly sharp. There you can see some diffusivity in this region just over here near the contact. And in terms of the rest of the outcrop, it's all dolomite. Okay, so this was part of the reason we wanted to run the study here, is because we wanted to see how well this would work on something that is almost entirely dolomite. So the wavelength, the carbonate wavelength imagery pretty easy to detect limestone versus dolomite, but what is it going to tell us about different dolomite phases? And will it be able to detect small differences in, say, magnesium? So looking at the dolomites, just to give you a bit more information on them, they are dominantly a bioturbated packstone. The burrows are horizontal. We're in the Cambrian here. You don't tend to see anything other than really horizontal burrows. There are some stylolites that tend to be focused on bedding planes. And in terms of the crystal sizes, they're ranging from about 50 to 500 microns. So not very coarsely crystalline. And the dolomites are not very well formed, more like a non-stoichiometric, non-rhombohedral dolomite. And the saddle dolomites within the breccia. So the breccia, you might have been able to see on the outcrop imagery that the breccia contains a lot of saddle dolomite. Some of these can get up to millimeter or millimeter scale crystals. I'm just going to switch this image for a second so you can see the difference between these. We have the large saddle dolomite crystals, and there is actually some quartz within this as well. Ooh, alphagenic quartz. And the saddle dolomite is, again, filling in what looks like mostly void space. So we see it between the clasts in the breccias, and we also see it concentrating along the zebra dolomites in the lighter zones. So if we take a look here, we have a clast. So this is very close up at the outcrop. We have basically a clast that's about 10 centimeters across. And then we have a zone of what looks like an altered replacement dolomite. So in this case, the clast actually displays some texture. We have some, just a little clast here of what looks like a cyanobacterial mats. And this is something that we see across the highway in an area that's just replacive dolomite. And we can see that texture of cyanobacterial mats that is preserved only in the clasts in this breccia. Now, the brown rim around the outer edge, this is something that may be a result of sort of a baked edge, if you like, from having higher temperature saddle dolomites and filling the void space. And then we have our saddle dolomites. And overall, if we take a look at the spectral profile, what we see is we have, oh, I think my slide changed on its own, sorry. Okay, so I'm just going to focus in on the compositional end of the spectral profile. And what we can see right away is we determined that the saddle dolomite was based on the spectra and the trough of this spectra. It is lower wavelength. And basically it's showing that it is getting close to the highest amount of magnesium that's detected. So basically the lowest possible wavelength for dolomite is 2315. And this is getting quite close to that. And then we have the calcite spectra is at 2330, which is where we're totally devoid of dolomite. We don't reach that, obviously, because all of this is dolomite, but the RD1 is showing quite a bit more calcium within the dolomite itself. So more magnesium, and that could be magnesium or iron within that SD profile. But overall, basically, we have less magnesium in the RD1 phase, and then slightly more magnesium in the RD2 phase, and the saddle dolomite tends to have the most magnesium based on the spectra. So for looking at compositional data, I just zoomed in on this interval here, but looking overall at the entire spectral profile can be helpful for determining the amount of crystallinity, or basically crystal size. What we're seeing in terms of what's on the left-hand side of this profile is that we have this peak. You'll see in the saddle dolomite, the very large peak that we see here. This is reflectance essentially. So this is a measure of how much light is being reflected back at us. And therefore could be thought of as maybe an indicator of crystal size, because a larger crystal is going to be able to reflect more light towards you. And we can see that with the RD1, there's almost no peak. This is a representative spectral profile that was taken in a region such as, sorry, I'm not sure why this is changing. This is taken in a region such as what I've shown in the picture there, and the pixels would have been grabbed to create sort of an average map. And we can see that the crystalline texture here, we have very fine crystals or no crystals in the RD1, nothing that's creating any kind of reflectance. And then we have the RD2, which is showing slightly more reflectance. So possibly the crystals have been neomorphos and they have increased in size in the second phase of replacement dolomitization. And then the saddle dolomite crystals are very large, which you can even see some of the saddle dolomite crystals in this photo. So not very surprising. And finally, this is the full image from the outcrop that we were able to detect. We have just a little gap there where we didn't realize that we hadn't overlapped, but you can get a sense of just how much detail is preserved in an image like this. Each pixel here would represent 3 to 5 centimeters in the outcrop. And basically the way that this was created, where we are showing those RD1, RD2, and the distribution of saddle dolomite, is through an analysis known as spectral angle mapping. So in spectral angle mapping, basically you are running statistics on the spectra and looking at the angle of reflectance and how intense that is. And it will look at the entire spectra itself as well, the compositional and more textural end, and run an analysis looking for similarities and differences in order to put them into groups. And finally, the way that we did this was we had basically 16 spectral groups that we analyzed. These were provided to me from Dr. Brevard and Dr. Fang. And I worked with Jack Stacey to go through these different groups and decide which of the spectral groups represented what phases and whether they should be grouped or whether they should be eliminated or split. So in the case of the 16 spectral groups, we had some that we eliminated. So anything that's in red here was eliminated because it, for example, was showing vegetation or fractures that we couldn't really, we think it might have been reflecting water, so just recent water and erosion. And then we also saw some that just didn't really show us anything, and it wasn't clear what it was representing. So out of the 16 groups, we eliminated 4, and then we grouped the rest together in such a way that represented our RD1, RD2, and saddle dolomite phases. Now, I'll just draw your attention to the last 4 here, EM13 through 16. These were all showing us they all represented the saddle dolomite phase. So I'll just have an image down here at the bottom, EM16, and you can see that there's some large clasts surrounded by a cement. The cement is highlighted in white here in this grayscale image, and that would be that EM16 showing what we thought was a middle sort of pore filling phase. And if we take a look at that breccia that I've shown in images before, we'll just zoom in on that from the whole data set. What was really surprising in this analysis, oh, sorry, these timings are, okay, there we go. So what was really surprising in this analysis was actually that we weren't sure, first of all, if we would be able to detect very easily between the replacement dolomite, saddle dolomite, and different phases of replacement dolomite. But also within the saddle dolomite, we weren't sure whether we would be able to see any difference within the saddle dolomite. But what was really interesting is those 4 spectral groups, if we take a look at those individually and split those out and assign them colors, we were actually able to come up with a cement filling map. So in this case, we can see if we zoom in on this image, that the zebra Dolomites over on this side, we have, oh my goodness, we have the pink phase, which is a pore lining phase. It's almost pretty consistently throughout the whole image, shown along the edges of all these void spaces. Then we have this blue and purple phase. And finally, in every case, the yellow would represent the final sort of pore filling phase in this breccia and within the zebras. And this was pretty surprising. So you can see the consistency in that image. And in this image here, where we have the very repetitive fill of the pore lining, pore filling, and then secondary and third pore filling phases. Now, if we take a look at the reference spectra, we again can look at the compositional side and the textural side. In the compositional side, what we see is that it's difficult to tell from this picture, but actually, I think you can probably see it. The yellow here would represent that final pore filling phase and is the lowest on the wavelength spectrum. And in this case, we have more magnesium in that central pore filling phase. And the one that is the pore lining phase actually has the lowest amount of magnesium or iron. Now, subsequent petrography has suggested that potentially these blue and purple phases could be grouped together. They are, I apologize for this, I don't know why the timing is going. Anyway, so those, basically, if we look at the paragenesis of this, we have the pink pore filling phase, and then the purple and blue, and then finally we would have the final fill highlighted with the yellow pore filling phase. And in terms of the composition, in terms of the texture, we can see that the pore lining phase has the smallest peak in the reflectance textural side of the spectra, and that the yellow pore filling phase has the largest crystal size. And the similarities between the blue and purple phases, you can see they are actually quite similar. And again, the petrography that we've done subsequently suggests that these might be actually combined. Okay. So overall, just to summarize the talk, we have the shales and the outcrop scans. In terms of the shales, I think some of the most valuable information you can get from the shales is continuous mineralogical data. Also, being able to image or calculate the TOC from the imagery and the spectral data is quite valuable, of course, to show the distribution. And you can identify sedimentary and biogenic structures, something I was particularly interested in telling the story of the deposition of the Duvernay, of course. And these structures would be otherwise undetected. So being able to detect these is, incredibly valuable because you would not be able to see them otherwise in some cases. And in the outcrop, we were able to image several distinct phases. We were able to decipher between replacement dolomite versus a pore filling cement dolomite, like saddle versus replacement. And what was really surprising was that in the saddle dolomite fill, we were able to see a differentiation of all the different saddle dolomite phases that have infilled this breccia and zebras, and that was very consistent throughout in terms of the different phases. We were able to detect their crystal size and the composition, seeing that there actually was a compositional difference and an evolution of the fluids from the pore lining to the pore filling phase. In both cases, I think the final thing I want to stress is that hyperspectral imagery, if you have access to it, is extremely helpful in determining a sampling strategy. So it's not often that we get to spend long periods of time in the field or at the core lab. And if you're able to use any kind of, there are portable imagers, then you can actually use that to develop a sampling strategy that will capture the heterogeneity in something like a shale that looks very monotonous, or in an outcrop that is entirely dolomite, in the case that we were doing on the cathedral. And finally, the acknowledgements. I just want to acknowledge the lab that I work with, the University of Alberta, and Drs. Benoit Rivard and Jialou Feng, who have taught me a lot about hyperspectral imaging and about spectroscopy. And also Professor Kathy Hollis and Jack Stacey from the University of Manchester, Tyler and Jesse, who helped with imagery at the outcrop from the Alberta Geological Survey, and finally the Alberta Parks for allowing us to work in the Kootenay Plains Ecological Reserve. Thanks very much, and I'm so sorry about the timing. I have no idea why that happened. I'm happy to take any questions you may have.
All right. Thank you very much, Hilary. That was a great presentation. It definitely seems like something we should be doing right now, since we can't really go to outcrops or the core lab and get lots of data from it. We've already got a couple of questions, so we'll get right into it. So the first one is from Warren Duncan at Murphy Oil in Houston, Texas. And his question is, are you using spectral libraries to classify the minerals, or have you?
Yes, so we use spectral libraries and also use literature basically to, for example, in the case of the carbonates, we would use some of the historical literature on specifically the spectra that represent different parts of the carbonate spectrum. And so a combination of both literature that was focused on particular problems like differentiation of carbonates, but the spectral library to overall differentiate between different phases of clay. Some of it ends up being a bit of trial and error, where we use the spectral libraries as a guide, but then, for example, something like the amorphous silica versus the crystalline silica, there was some information about that in the literature, but in terms of understanding the single peak to double peak spectrum, sort of when we have that shoulder appear, we wanted to understand how that related to different different silica phases. And so we did some testing by grabbing samples from the museum and from laboratories and that had previous analysis, and tried to test whether we could differentiate between different species. So it's kind of a combination of all those things.
All right. The next question is from Tim Cullen at the University of Bergen. It says, thanks for the great presentation. A couple of quick questions on the logistics of core analysis. So for first question, are there any particular core preparation routines that need to occur prior to hyperspectral analysis, or restrictions? Examples, core slab thickness for hyperspectral analysis to be effective. And then his second question is, regarding TOC analysis, how susceptible to errors is this in calcareous mudstones? Is there a concern if you were to carry out such investigations in marls, for example? Thinking in comparison to troubles that occur with other analytical methods for TOC in very calcite-rich but still clastic rocks where we might be interested in TOC.
Right, okay, so I can definitely answer the first one. The second one, I have some suggestions, but the first one, I would say there are some ways you can prep the core for sure. Core thickness doesn't matter because we're just looking at the very surface. We're only looking at reflectance. So while there is a very minimal, like millimeter scale penetration into the core in terms of what kind of data we're getting back, mostly it's just superficial. So actually, given that, the best way to prep a core is to ensure that all dust is removed. We generally clean these cores ahead of time because they are so incredibly dusty, especially after being transported from Calgary to Edmonton. So we clean the core, and sometimes we'll clean with water, but in fact, one of the best methods we found is using an eraser and erasing some of the core. So for our high resolution imagery, we would have taken an eraser and run it over the core, just a standard eraser. And that helps clean off all that dust. And we have tried imaging cores that have been wet before, but the water feature can be a little overpowering. So if you do wash the core, you want to ensure there's a significant, enough drying time, like overnight, for example. Well, overnight's good enough in Alberta because it's very dry. You might need more time in England. But giving it sufficient time to dry is important as well, because the water feature is quite strong and can interfere with the other spectral signatures or make them more difficult to detect and isolate. As for your second question, that is a good question. And in terms of the success rate of the prediction of TOC, I think the results have been pretty consistent in terms of the predicting TOC in carbonates versus more clay-rich intervals. For example, the Duvernay is present also it's present throughout the basin, but in that first image, I showed the, I'll just go to it now. In this image here, I showed the different reefs and the distribution of the Duvernay. This series of barrier reefs that form in a line here separates what we know as the East Shale and the West Shale basin. And the East Shale Duvernay is completely different than the West Shale Duvernay in terms of carbonate content. It's surrounded by carbonate platforms, and therefore there's a lot more carbonate shed from those platforms and a lot more carbonate incorporated into the Duvernay. In fact, the drilling strategy and approach for drilling East Shale Duvernay is totally different than the West Shale. And that being said, we ran some TOC analysis on the East Shale Duvernay and found that the analysis of TOC that we ran in the lab versus from our samples versus the predicted TOC still had a fairly good agreement. I couldn't give you the exact figure, but it is definitely a great way to detect TOC in terms of relative amounts, and you can get some numbers from that, and the consistency with lab analyzed TOC samples is pretty good. But don't quote me on that. That's just one example that I've done. I would actually look at Benoit's 2018 paper because he talks a lot about the detection of TOC in that paper, and that's a technique that their lab developed. So that'd be my recommendation.
All right, next question is from Stephen in Wales. Fabulous webinar, thanks. Do you know for any instances where this has been used on mixed volcanoclastic carbonate lithologies?
Ooh, the short answer is I don't. But I'd love to see that. I actually think it would be wonderful to do a ton of different analyses, you know, to create a library almost of this technique. And with the technique becoming more available in terms of the costs coming down, I think that's something that could potentially happen over the next few years. The other really interesting thing is that we've put in, there are some people, and we've been trying ourselves to get a drone that would be mounted with the hyperspectral equipment. And you know, this technique originated as a remote sensing technique it was mounted on the bottom of planes that would do flyovers in certain regions. And now it's been adapted for this, you know, using the outcrop scanners and core scanners. But putting that a hyperspectral sensor detector on a drone would just be fabulous, and pairing it with LiDAR is what we hope to do. But of course, the issue always comes with drone work, that if you mount $300,000 worth of equipment on a drone and it crashes, that can cause problems. So yeah, I don't know of any, Stephen, but I think that's a great idea and something I'd love to image.
All right, the next question is from Stan in Houston. Fascinating, Hillary, thank you. Today you have mostly spoken about the use in carbonate/shale systems. Are there any examples of effective use in more coarse-grained siliciclastic systems?
Yes, so actually there is a student that Benoit had, Michelle Spectra, and I'm trying to think when her papers came out. It's going to be somewhere in the range of 2016, 2018. They actually looked at the application of this technique in the oil sands in Alberta, which are coarse siliciclastics. And the really interesting thing about their study is they were able to completely remove the effect of having bitumen soaked, like entirely bitumen soaked core. So if you look at oil sands core, I don't know if you've seen any oil sands core from Alberta, but it's black. It's core that you can't work on unless you're in a well-ventilated room, because it stinks, you know, so badly, and it'll give you a headache. But what's really interesting is they were able to image the core and remove the effect of the bitumen, and able to detect a ton of sedimentary structures, biogenic structures, And it gave them the ability to have a whole interpretation of a core that you would never normally have without totally having to clean it. And it also highlighted, very subtle differences in mineralogies that can show sedimentary structures in a level of detail that you might not normally get from something that's very quartz rich.
Excellent. Another question from Warren Duncan again. You mentioned the difference between the scanners and the handheld. Can you replicate the scanner results with the handheld? If not, how much is lost? Are you using spectral libraries or did you acquire your own spectral samples? TOC analyses, did you use secondary absorption peaks? If so, did you determine those peaks?
Okay, so the first question, sorry, I got a little lost there. What was the first question?
Can you replicate the scanner results with the handheld? And if not, how much is lost?
Okay, so in terms of the data that I have been showing, which is mostly imagery, that would not be what you would get from the handheld detector. But in terms of what was shown in the image with the TOC, which I'll just go to that now. So something like this. This TOC weight percent is actually something you could collect with a handheld sensor. So you get the same spectra. In fact, the handheld sensors are more precise in terms of the spectra that they will provide. And so if we have questions about some of the, if we have messy spectra with noise and various things, we would actually take the handheld sensor and apply that in specific regions to clarify what we're seeing and make sure that we've accounted for any noise associated with imaging from a greater distance. Detecting from a greater distance. Hyperspectral imagery is basically spectral data paired with high imagery or high resolution photography. And so you wouldn't really be able to replicate exactly what I've shown in terms of the imagery with grayscale or RGB images, but you would definitely be able to have obtain mineralogical data. You would still be able to model TOC, and it would be more similar though to when you're doing spot analysis through sampling. Of course, it's quicker, right? So you can actually, just like an XRF, you can run through with a hand analyzer, or there's some that are mounted, but it wouldn't produce the imagery. So you can take high resolution photos. For example, I know of a company that has a hand sampler mounted to an automated core logger, and it takes images with each spot, but you wouldn't end up with the same data where you have pixels that actually contain the spectral data. So it wouldn't be as continuous. What was the next question?
For TOC analysis, do you use secondary absorption peaks? If so, did you determine those peaks?
I think, sorry, but that would be something that's getting out of my range of expertise. So in terms of the way we work, a lot of the time especially with the TOC, I was not really involved in the production of the model that we use for TOC. But if you wanted more information on that, you could contact Dr. Benoit Rivard. I'm kind of more on the mineralogical side and the modeling of TOC. It is covered in detail in that paper that I referenced, so it might be worth checking that out, but I apologize, I wouldn't be able to give you any solid information on that.
All right, our next question is from Tobias Kurt from NGU Trondheim. Nice presentation, Hillary. Has some validation done on the outcrop hydrospectral mapping? What and how was it done?
Good question. So we are still validating some of the results that we found. We've done an initial petrographic analysis and also ChemScan imagery to verify some of the differences that we've seen in terms of the composition of the spectra. We are looking at sort of, if you remember back to, hang on, the spectral profiles here. What we've tried to do with the ChemScan mapping so far is to look at the composition of the crystals that we see in transects across the Saddle Dolomite. And in fact, we do see verification in terms of the yellow phase being more magnesium rich. In fact, it is more magnesium iron rich. And the pore lining pink phase actually is from the ChemScan data and microprobe data we have so far shows that we can validate this and say that it's more magnesium or iron poor. So we've started doing some analysis like that. What we are also hoping to do is we've subsampled in a number of these regions and then scanned the hand samples. And we hope to run some very detailed geochemical analysis on that. We have Kathy and I have a new PhD student, Cole McCormick, who's working on some of this for his PhD.
All right. The next question is from John Faithful in Scotland. Spectral processing tools, how open are raw data sets? How reliant on closed or priority software are you? Or are they open community tools that can be retrospectively applied to data sets from diverse sources?
That's a good question. So as far as I know, because I've spoken with Dr. Fung, who I work with on all of this very closely, he has told me that there are people trying to develop software that's going to be open source that would be able to handle this kind of data. At the moment, all of our data is processed using Envy, which is proprietary software. I mean, you can purchase it, but it's quite expensive. So it's a little bit limiting at the moment. But apparently, I don't know, according to Dr. Fung, he says that he's aware of people who are trying to provide open source software. There might be someone, actually, Tobias Kurtz, the person who asked the last question, he might be able to comment on that as well. I'm not aware of any free software to analyze the data at the moment.
All right. Well, I think we'll wrap that up. Thanks again, Hillary, for your great talk. And please join us next week at the regular time, 4 p.m. UK time, when Teresa Knoll will be talking about limestone marl alterations. Hope to see you then.